Thank you for having me here today. I'll be talking about our new metagenome profiler called sylph. This talk will be about metagenomics, in particular shotgun metagenomics, so not 16S. And in general, what we do is we have a microbiome sample that we're interested in studying, like the gut. We sequence it, and really we want to ask, what is in my sample? That's the key question. And we can do this through a couple of methods. One is assembly, so you can de novo assemble the sequences, and this is great because you can discover new organisms. You can do profiling, which is using a database to infer taxa abundance or presence. And this is useful when we can't do assembly. And this talk, I'll be focusing primarily on profiling, which is the database approach. So this is the standard idea that people use in profiling. This is the first thing that you would kind of think of, which is you would map reads to genomes, and then you calculate some sort of relative abundance by classifying the reads, right? Some reads go to E. coli, some reads go to Salmonella, and I can kind of infer the presence of those organisms. Obviously, I'll have some unclassified reads as well because these databases aren't perfect. So this is a great idea. Why build a new metagenome profiler, right? These aligners, they work really well. This should be no problem. The problem is that classifying reads is actually very hard. Um, these databases are becoming really, really big. You can have hundreds of thousands of bacterial archaeal genomes, and this is really expensive to do. It takes a lot of memory and time. And also, false positives when you classify reads are kind of inevitable because reads are short a lot of the times, and you can't classify them, right? You'll end up getting false positives. So that's why we are introducing a new metagenome profiler, and this is by something called k-mer containment. Um, this is out in bioRxiv. It's our new profiler. It's called sylph. So the idea is to classify genomes against reads instead. So you take each genome and you look at it, it's contained in your reads one at a time. And the way we do this is through a couple of steps. The first one is called k-mer sketching. So k-mers are these short substrings of a larger string of length k. And sketching is when you sample some of the k-mers. You don't use all of them. You undersample or subsample some of them because that speeds up your computations. And the first step of our program is to do k-mer sketching on both the reference genomes, your database, as well as your reads. And we use a technique called frac min hash. If you're familiar with minimizers, it's the same idea. If you're not familiar, then just think about some way we're subsampling the k-mers. And we subsample about 1 out of 200. So it speeds things up by 200 times. And the next step is called k-mer containment. I have my reference genome, I break it down into k-mers. I have my reads, I break it down to k-mers, and then I just compare these sets of k-mers. I can use intersections to see how contained my genome is in my reads. And there's a important connection to average nucleotide identity, or ANI here, that was already discussed before. If you have two strains that are 99% similar, they'll have obviously some base level differences. That means their k-mers will also differ. And the fact that their k-mers differ, you can use this. The upshot is that you can estimate ANI by counting k-mers and just seeing how much are shared. And this is really nice because counting k-mers is really, really fast, whereas alignment, which is how you would usually do this, is very slow. And in practice, our k-mers are around 20 to 32 in length. Of course, ANI is just for genome to genomes, but we're comparing metagenomes here. So I want to find out, is my genome contained in my metagenome? And we could the thing about that formulation I showed before is just I'm just doing intersections of k-mers, and this generalizes the metagenomes. Obviously, there are some issues here about what does this really mean. It's kind of like a nearest neighbor sort of ANI, but it works, and you can just extend the formula. And this is really nice because we care about ANI because it's delimiting. It delimits species. Um, in this field of microbial genomics, we use this kind of heuristic threshold now, which has become quite popular, which is that if you're more than 95% ANI, two genomes, then you're the same species. Some people have argued back and forth, but it's become kind of a standard to use this. So because we can calculate the containment ANI of that genome to the metagenome, I can use this to estimate, is it present at the species level? So this is very simple, representation of how sylph works. You kind of just try one genome at a time and see if our previous formula says it's present at species level. So that's fantastic, but there's a very big problem here. It's called the low coverage problem, and this is our main innovation. It turns out, well, I don't have just a metagenome that's complete. I sequence the metagenome, right? And because I'm sequencing the metagenome, I'm going to miss stuff if I don't sequence everything to really, really high depth. For low abundant organisms, you can see the transparent k-mers there. Um, we just didn't sequence them. We got unlucky, and we didn't sequence the DNA. So this formula that we used before, it doesn't work because that B assumes that we have the entire genomes available, but we don't. We have a sequencing of the genomes. So this method fails. And I want to show you how we can still recover some information now. So on top there, I have a reference genome. The reference genome, there's no sequencing for the reference genome. I just have my genome, right? And I have the k-mers for my reference genome. On the bottom, I have a metagenome, which is represented by one genome here. And it has some X's, which are like base level differences to my genome. Okay, but the metagenome is sequenced. If I look at these k-mers, this first k-mer here, it's got coverage too, because it's sequenced twice in my metagenome. So that's fantastic. But this k-mer over here, it's got coverage 0. It's sequenced 0 times. Why? Because there's a difference. This k-mer is not sequenced exactly. Like a erroneous or mutated version of it is sequenced. So it's got coverage 0. Whereas this k-mer as well, it also has coverage 0. But the reason it has coverage 0 is because I got unlucky and I just didn't sequence it. Okay? So you end up getting these k-mers that drop out due to mutation and sequencing. So what sylph does is it does a statistical adjustment for this low coverage problem when you have like k-mers dropping out due to sequencing. So I want to show you some intuition here. The first thing up top is a histogram of all of those coverage counts. And you'll see this kind of nice like Poisson part, but you also see a zero inflated part. Why? Because you'll have zero inflation due to mutation. Those mutations cause the k-mers to have like 0 coverage or drop out. But you'll also have some zeros due to sequencing. And sequencing follows a Poisson distribution. This kind of a classical stochastic assumption that we make. But the point is nucleotide identity doesn't care about my sequencing. Nucleotide identity just cares about mutation. So what I really want is which zeros are due to mutation. And I need to deconvolve this. And this is what sylph does. So the first step of sylph is to infer that Poisson part by looking at this distribution. And then you readjust it based on this Poisson part. You can readjust to find out what zeros are actually due to mutation. And then this naive formula that I showed before, that's the one that's crossed out. This doesn't take into account sequencing, but our new formula that we derived takes into account sequencing and is able to give a better estimate, which I will show now in our results. This first experiment is I take a genome and I just sample reads. I just simulate reads from the genome. It should have 100% ANI, my sampled metagenome, but it doesn't. You can see that there's this kind of lag, this underestimation that occurs. The purple one is using that naive containment that I talked about that doesn't take into account sequencing. But the other ones, I use the statistical adjustment at various sampling rates. This is the sampling frequency of the k-mers. And you can see that it's adjusting to the true 100%, which is what I actually want. So it starts to work pretty well, even at quite low coverage, like 0.03 X coverage. And well, we can use it for profiling. This is what I talked about before. And in particular, you can use it to estimate abundances. I didn't talk about how to use it to estimate abundances, but it's possible. And what we do is we're operating on a data set. It's called the CAMI2 data set. It's a standardized data set people use in metagenomics for testing things. And for species level profiling as a classification task, we do pretty well, very comparable to other methods. And the abundance estimates are quite good as well. But I think what's more interesting is how fast and efficient our method is. Recall that we're really sampling these k-mers and not using a lot of information. So we make it really, really fast. It's a couple orders of magnitudes faster than other methods, and it uses a lot less memory as well, right? So this database is about 30,000 genomes. And Kraken, if you're familiar, Kraken's a very popular tool. It's already using 130 gigs of memory, but we're only using about 4. So it's scalable. Final application I want to discuss is really interesting task that other folks did with our tool for contamination detection. So if you look at public sequencing data, there's metadata available, of course, but this metadata may not always be accurate. And furthermore, if you take even isolate sequencing reads, there'll be contamination in those reads. So this one solution that people came up with is you just run a metagenome profiler on isolate reads, and it should pick up contamination, right? If you have only E. coli, but then you run a metagenome profiler and you find out that like 50% of the reads are something else, that's very helpful information. So in this recent paper, called All the Bacteria. These folks assembled and quality controlled a lot of genomes. They did a contamination check and assembly for every single SRA data set for bacterial isolate illumina whole genome sequencing. And you can try to guess how many data sets this is, but uh, they analyzed 2 million sequencing data sets. And for the contamination part, checking contamination, they use sylph to make sure that the metadata aligned and there wasn't too much contamination. And they found that sylph was accurate, faster, and required less RAM than previous tools for even 85,000 genomes. So this is one of the GTDB data sets, if you're familiar with the data set, which is 85,000 genomes and only took 10 gigabytes of RAM. So just to recap, we built a new metagenome profiler. It's called sylph. And there's really two main ideas. The first idea is to kind of classify genomes against reads using these k-mers, and it works pretty well. And the second idea, which is really important, which is our main innovation, is to use these coverage-aware statistics for analyzing your sample and estimating this nucleotide identity that gives us accurate species-level profiling. So of course, there are some drawbacks to sylph. It can't just be 30 times faster and more precise and not have any drawbacks. It doesn't classify reads. So if you're familiar with tools like Kraken, or if you just take read aligners, those classify each read. But this one, sylph does not classify reads, and some tasks require classifying reads, right? If I only give you like 2 reads, then you can't really run sylph. Another drawback is that we require species level representatives in our database because we really only check species level presence. But because it's so efficient, you can incorporate massive databases, you can incorporate hundreds of thousands of genomes, and this alleviates the issue somewhat. So in conclusion, our paper, the title is up there. It's available on bioRxiv and it's in review right now. I'm Jim. I'm a 5th year PhD student, and this is work with my advisor William Yu, who's an assistant professor at Carnegie Mellon. And this work is supported by NSERC and the Canadian government. And yeah, thank you very much.
Great. Questions for Jim? Right here.
Yeah, it's interesting right now. I don't know if this is just a problem with transcriptomics, but in our experience, when we work with k-mers, we end up with millions, billions of k-mers, and making it impossible to load it all memory.
Yeah.
What was your strategy to deal with all that?
Right. So the big thing is the sampling. Because we're only taking about 1 out of 200 k-mers, you can imagine you just reduce your memory cost by 200 times. So that's the main thing. Another thing that helps as well is that these metagenomic read data sets, they're not too bad. They're not the biggest data sets if you analyze one sample at a time. Yeah.
Thank you.
I had a question similar along the same line since you mentioned Kraken. And Kraken uses k-mer as well.
Mhm.
What are you doing different from Kraken?
So Kraken. Oh, so the question is, Kraken also uses k-mer. Kraken is a very common tool people use for profiling. What are we using differently than Kraken? And what we're doing differently is that Kraken classifies every single read, and they look at how reads map to the databases. What we do is we take k-mers for both reads and the genomes, but we look at how genomes are kind of contained within our reads. So we're kind of like doing an inverse kind of problem to Kraken, and we're also sampling way more aggressively. Kraken samples about 1 out of 8 k-mers. We sample 1 out of 200.
Good question back there.
I wonder how you came up with the number 200.
Yeah. Yeah.
Is that some part of experimentation? Does it depend on that or some kind of other parameters?
Yeah, great question. I think there's 2 things. No. Yeah. So we're sampling about 1 out of 200 k-mers. How did we come up with this number? Did we do it through experimentation, or is there a theoretical reason? There's 2 things. Experimentation for sure. It works pretty well at 200. It worked okay at 100 as well, but we got a 2 times speed up, sampling 1 out of 200 instead of 1 out of 100. Another thing is that reads are about 150 base pairs for short reads. And what we found was that we actually wanted these k-mers to be a little over 1 read spread apart. And there's some theoretical reason with the statistical model that you want that. So a little bit of both.
Great. Other questions? Maybe one last one.
Yeah. So do you do anything to try and ensure that the k-mers that you pull from each genome are unique to those genomes? Like, do you like try to specifically pick unique ones?
Sure. So no, we don't actually do anything to ensure that the k-mers are unique. What we do is we pick K to be around 31 in practice, which turns out for bacterial genomes and archaeal genomes to be relatively specific. That won't work on human genomes, but it just works okay here. We could use something like that. That would take a little bit more processing, but some people have done something that like what you suggested. Yeah.